Hello guys, in this part I want to talk about the cardiac conductive system for you. The conduction system of the heart can initiate and conduct the electrical events, and it can help proper timing contraction. In our heart, we have a specialized muscle cell which have the action potential, but they don't contract themselves. Their activity is influenced by autonomic nervous system, and in this part, I want to talk about this conduction system of the heart. We have a group of cells in different parts of our heart in cardiac conduction system. The first group is called SA node. The SA node or Cyanoatrial node can initiate the heartbeat for us. Actually, this is the pacemaker of our heart, and it's located in the posterior wall of our right atrium, in the opening of superior vena cava. The other group of cells are called Atrioventricular Node or AV node. The location of AV node is in the floor of right atrium, and it's very near to the right AV valve. The next part is called atrioventricular or AV bundle. The other name of atrioventricular bundle is His bundle or bundle of His. It extends from AV node and come down in interventricular septum, and then it divides to right and left branches. And we have some Purkinje fibers which extend from the right and left bundle at the apex of the heart, and they come down in the lateral wall of the heart and bring the information of contraction in all parts of the heart. Now in this picture you can see all of these structures. The first part is called Cyanoatrial node or SA node. You see that it's in the posterior wall of our right atrium near the opening of superior vena cava. The other node is called AV node or atrioventricular node. It's located near the right AV valve in the floor of right atrium. Then from this AV node we have a bundle which we call it AV bundle or His bundle. It enters the interventricular septum. It passes about 1/3 of interventricular septum, and then it divides into two branches. One of them is called right AV branches and the other one is called left AV branch. Then they come down to the apex of the heart as you see here, and after that they go up in the lateral wall of the heart as Purkinje fibers or Purkinje cells, and they can bring the contraction information to all parts of the heart. These parts, which you can see them in yellow color, can initiate the action potential and contraction, but they don't go to contraction themselves. The innervation of the heart. We have a cardiac center in our medulla oblongata. In this part, we have two important centers. One of these centers is called cardio-acceleratory center. It helps our heart to beat more, and the force of contraction of our heart increase when this center becomes stimulated. And the other center is called cardio-inhibitory center. This center can make the heart rate lower, but it doesn't work on the power of contraction of the heart. These two centers can receive their signals from two important receptors. One of these receptors is called baroreceptor, and the other one is called chemoreceptor. Receptors in the next slide. These receptors are located, let me bring the slide for you. These receptors are located, as you see here, in the aortic arch, and also the other receptor is located in the bifurcation of one important artery in our body, which we call it common carotid artery. I will talk about this artery in the blood vessels for you. But the location location of these baroreceptors and chemoreceptors are in aortic arch and in the bifurcation of common carotid arteries. The baroreceptors can check the pressure of the blood and the pressure of oxygen, the pressure of carbon dioxide in our blood, and the chemoreceptors can check the pH of our blood, the chemical composition of the blood, and all the times these two group of receptors can monitor our blood and send the information to the medulla oblongata as you see here. Sometimes we have activation and the stimulation of cardio-acceleratory center, and sometimes we have activation and the stimulation of cardio-inhibitory center. Let me go back to this slide. So, and after that, when all of this information go to medulla oblongata, the medulla can send signals by two pathways. One of these pathways is sympathetic and the other one is parasympathetic pathway. And I will talk about them in the next slide for you. And both of them, sympathetic and parasympathetic, can modify our cardiac activity. Pay attention. They can only modify it. They can only change it. They cannot initiate it. And you need to know that when we have a frog, for example, and we put it in a tray, we kill the frog and we separate the heart from the body and put it in the other tray. The heart beats for about 30 minutes. It means that the heart can beat without any connection to our body and to our nervous system. So inside the heart, we have a system which can generate the action potential and it can generate the contraction. And only the nervous system, the sympathetic and parasympathetic pathways, can modify and change the power of contraction and also the rate of our heart. The parasympathetic innervation, as you see in this slide, can decrease our heart rate. So the parasympathetic can decrease the heart rate. It starts from the cardio-inhibitory center in our medulla, and the vagus nerve can bring this information to our heart. The right vagus nerve can innervate our SA node, and the left vagus nerve can innervate the AV node. The sympathetic innervation can increase our heart rate and force of contraction. So you see that when you are in stress, when you are doing workout, when your sympathetic nerve becomes stimulated, your heart rate increase and the power of contraction goes up. The sympathetic innervation starts from the medulla cardio-acceleratory center, and it comes down by nerve thoracic number 1 to thoracic number 5 of the spinal cord, and it extends to SA node, to AV node, myocardium, and coronary arteries. The function of this nerve is increasing heart rate, power of contraction, and also the other function, which is very important, is it can increase the coronary vessel dilation. So we have dilation of coronary arteries, and they can bring more blood with more oxygen and more nutrient for our cardiac muscles. And again, in this picture, you can see the baroreceptors and chemoreceptors in the aortic arch and the bifurcation of common carotid arteries. You see that the information all the times go to our medulla. Sometimes, as you see here, the cardio-acceleratory center become active, and from T1 to T5, this sympathetic pathway can bring the information to our SA node, to our AV node, to the cardiac muscles, and to the coronary arteries. And when the parasympathetic nerve becomes stimulated, then you can see that we have a right and left vagus nerve. The right vagus nerve can bring information to the SA node, and the left vagus nerve can bring the information to the AV node, and they can decrease the heart rate. but the parasympathetic innervation don't work on the power of contraction of our heart. In this part, you can answer these two questions. Which of the two autonomic nervous system divisions have an effect on the force of heart contraction? And what is the cardioinhibitory center and where this center is located? The heart stimulation and the heart contraction involves two major events. One of them is the conduction system of the heart initiate and propagate the action potential. And we have cardiac muscle cell which can receive all of this action potential, and they start to fire and go into contraction. This process happens first in the atria and then in ventricles. It means that first two atria go to contraction, they become fully contracted, and when they go to contraction, they can send the blood through the right AV valve and left AV valve to right and left ventricle. At this time, our ventricles are in relaxation. When the atria send all of their blood down to the ventricles, the atria become relaxed, and at the same time, the ventricles go to contraction. So the contraction of these two parts are not at the same time. And I talked about it before for you in the fibrous skeleton of the heart, which can insulate our heart. And first, the two chambers which are located superior go to contraction, and after that the ventricles go to contraction. Now I want to talk about the physiologic process which associated with the heart contraction, and I want to talk about the cells which we have in SA node, AV node, AV bundle and bundle branches, and also the Purkinje cells. First, you need to know that these cells are completely different in their shape and in their properties when we compare them with cardiac muscle cell. As you remember from our previous video and previous part, the cardiac muscle cells are elongated in their shape. They have branching on both ends, as you see in this picture, and they can show striations here. And they have one or two nuclei which are located in the center of the cell. But the nodal cells, as you see here, doesn't have this characteristic. At the embryonic period, about 1% of the cells which want to make the heart change to these nodal cells. As you see in this picture, they don't have a striation, they have a regular shape, they contain a less myoglobin than the cardiac muscle cell, so their color is lighter. They contain a very large amount of glycogen inside, and they have the oval nucleus which is located in the center of the cell. These cells have many channels in their cell membrane, and by these channels they can initiate the action potential. The action potential first generated in the SA node. Then it comes down to the AV node, as you see here, by different routes. It comes to the AV node, and at the same time, due to presence of gap junctions between cardiac muscle cells, this information of contraction can easily distribute in both atria, and both atria can go to contraction, as you see here by green color. The contraction starts from the superior parts, and gradually it comes down to the inferior part of our atria. So we have a spread of action potential from SA node to AV node. After that, we have a delay for transmitting of this information in the AV node. Then it goes down to the AV bundle, bundle branches, reach the apex of the heart, and then goes up on both lateral wall of the heart. And here you can see the cardiac muscle cells. I told you that in the cardiac muscle cells, we have a branching pattern, we have striation, we have central nuclei, sometimes one and sometimes two nuclei, and we can see a striation here. Between the cells, we have intercalated disc, and the intercalated disc contains gap junctions and desmosomes, and through the gap junctions, the information of action potential can go cell by cell, and all parts of our heart can go to contraction at the same time. When the when the action potential reach each cardiac muscle cell, our cardiac muscle cell, which contains sarcomere, as you see here, they thin and thick filament can slide over each other, and the size of our cell becomes shorter and shorter. And this is the meaning of heart contraction. Now, I want to talk about the action potential generation in our nodal cells at rest. The nodal cells at rest. The nodal cell in SA node can initiate the heartbeat. They can go to depolarization automatically, and they can generate the action potential. The resting membrane potential in our nodal cell is about -60 millivolt. They have different pumps and different channels in their cell membrane. One of these pumps is called sodium-potassium pump, one of them is calcium pump, and they have some leaky channels. The channels which we have here are voltage-gated channels. We have a slow voltage-gated sodium channel, fast voltage-gated calcium channel, and voltage-gated channel for potassium ion. And in the next slide, you can see all of these channels. The first channel here is the slow voltage-gated sodium channel. The other one is the fast voltage calcium channel, and the other one is the voltage-gated potassium channels. And the difference between outside and inside during rest is -60 millivolt. The sodium channel here is always open, and sodium ion can come to our cell gradually. What happens when one positive ion comes to our cell? When positive ion come to our cell, inside the cell become more positive. So we can move from the resting membrane potential, which is -60 millivolt, up and up until we reach the threshold level. When we reach the threshold level, then let me draw the graph here for you. Imagine that this is -60 millivolts, and the sodium ion come to our cell, and inside the cell become more and more positive. When we reach the threshold level, which is here, then the calcium channels become open, and calcium ion come to our cell very quick. Because of this quick entrance of calcium ion, we call it fast voltage-gated calcium channels. Calcium ion come to our cell, and our cell go to depolarization. After that, when we reach zero here, when the difference between outside and inside becomes zero, the calcium channels become closed and potassium channels become open, and we have repolarization. So this is the graph of action potential which we have for our nodal cells. So the SA node cells can exhibit autorhythmicity. It means that it can do firing automatically. We have a slow voltage-gated sodium channels. They become open, sodium ion come to our cell, and we reach the threshold level. The threshold level is -40 millivolt. After that, the fast voltage calcium channels become open, calcium ion come to our cell, and membrane potential change reach about somehow above 0 millivolt. And then you have repolarization, calcium channels become closed, and voltage-gated potassium channels become open, and potassium come to our cell. And again, membrane potential reach back to the resting value. And again, when we reach negative 60 millivolt, the voltage-gated sodium channels become open, and the next action potential created here. Here you can see this graph, the graph of action potential in Nodal cells. Here we have negative 60, which is the resting membrane potential, and in this part, sodium ion, as you see here, come to our cell until we reach the threshold level, which is negative 40 millivolt. When we reach this level, the fast calcium-gated channels become open, and we have depolarization until we reach about 0. And then the potassium channels become open, the potassium ion leave our cell, and inside the cell again become more and more negative, as you see in this picture, and we reach negative 60 millivolt, which is the pacemaker the resting membrane potential. And when we reach negative 60, again, sodium channels become open, and the next action potential is created. At rest, one SA node action potential starts about 0.8 seconds after the last. So if we have this time for a generation of action potential in our SA node, the heart rate or the heartbeat becomes 75 beats per each minute. The SA node can fire faster, and it can beat about 100 per each minute. But in our body, we have a vagal tone. The vagal tone means that the parasympathetic activity have its very high impact and effect on our SA node, and it can keep the resting heart rate slower than 100 because it wants to reserve energy in our body more. Again, in this picture, you can see the SA node activity. During reaching threshold, only the voltage-gated sodium channels are open, and sodium ion can come to the cell. Here, the cytosol is upper, and the interstitial fluid is lower part. Then, when we reach the threshold level, the fast voltage-gated calcium channels become open, and we have influx of calcium ion inside the cell. Inside the cell become more positive from negative 40 to 0. And after that, we have repolarization. The fast voltage-gated channels become closed at when the difference between outside and inside becomes 0, and then the potassium channels become open, and you see that the potassium ion leave our cell again until the membrane potential reach negative 60 millivolt. Here in this slide, you can see the comparison between our nodal cells and neurons. The similarity of both of them is they can fire and generate the action potential, but we have some differences between them. In our neurons, we need a stimulation for the action potential. When the stimulation comes to the neuron, then some channels become open, some channels become closed, and we have generation of action potential. But in the nodal cell, we don't need any stimulation. They can go to action potential automatically. Then the nodal cell don't have a stable resting membrane potential. They exhibit a pacemaker potential when there is sodium channels become open. So immediately when our cell reach the resting membrane potential, the cells become open and we have entrance of sodium ion inside the cell. But we don't have this feature in our neuron. Let me draw the action potential of the neuron for you. If you remember, we have resting membrane potential, then we have depolarization, repolarization, hyperpolarization, and then again resting membrane potential. But in the nodal cell, as you see here, we have first reach threshold, then we have depolarization, repolarization, and immediately when we reach the resting membrane potential, the other wave start. So we don't have a a stable resting membrane potential. And in neuron, only we have entrance of sodium ion as the positive ion, which works here. But in these parts, as you see in this picture, we have one other ion, which is the calcium, which is important, and it can come to our cell and make our cell become more positive. After starting at SA node, the action potential is spread. The action potential is distributed through the atria and it reach, as you know, to the AV node. The excitation can travel via gap junctions and the two atria contract together. Action potential has delay at the AV node. AV nodal cells are slower because of they are smaller in their diameter and they have fewer gap junctions. So they are smaller and they have fewer gap junctions. And we have insulation of fibrous skeleton. So it's like a bottleneck here. And we have the only path here, and it can make a delay for the spreading of information. So we have the delay, which help the ventricles to fill before they go to contraction. And the action potential can travel through AV bundle to the Purkinje fibers. The Purkinje fibers are very similar to the nodal cell, just they are a little bigger than the nodal cells. And the action potential can spread in all parts of our ventricles after that. Gap junctions can allow the impulse to spread through the cardiac muscle fibers, and cells of two ventricles can go to contraction automatically. Now look at here in this picture, you can see all of these events. You can see the generation of action potential in the SA node spreading into atria, and the red color shows you the contraction. You see that when all parts of the atria go to contraction, then the AV node can start sending the information to the AV bundle. It goes to the AV bundle and then bundle branches. As you see in this picture, it comes to the bundle branches, it goes down until it reaches the apex of the heart, and when it reaches the apex, then the contraction starts in our cardiac, ventricles, and you can see that the wave of contraction starts from the apex and come up. Why we have this wave from down to above? It's because of it wants to go to contraction in this way to send the blood to pulmonary trunk and aorta, which are located in the superior part of our ventricles. Specialized feature which associated with our ventricles. One of them is the Purkinje fibers. The Purkinje fibers are larger in diameter than the other cardiac fibers. The action potential are very rapid in these fibers, and it ensures ventricle contract at the same time. The other feature which is important is the papillary muscle. I hope you remember the papillary muscle. The papillary muscle, which we have 3 in right ventricle and 2 in left ventricle, are some conical muscles which are attached to chorda tendini, and the chorda tendini are attached to the valve, right AV valve and left AV valve on the other side. When first, you need to know that the papillary muscles are the first part of the ventricles which go to contraction. When they contract, they make the chorda tendini become taut. When they become taut, they close the AV valve. And when our ventricle go to contraction, the only way where the blood can go are the pulmonary trunk from the right ventricle and aorta from the left ventricle. So when we have any problem with our papillary muscle contraction or chorda tendini deficiency, then this valve cannot completely close and we have regurgitation of blood from ventricle to atria. Don't want it. The other feature which is very important is a stimulation begin at the heart apex, and it wants to ensure blood efficiently ejected toward atrial trunks. And we have some problems like ectopic pacemakers. The SA node is called pacemaker of the heart. Sometimes in our hearts we have other spots which work as pacemaker and they can generate the action potential. And conduction system cell other than the SA node cell, they also have this ability to spontaneously go to depolarization. They become depolarized at a slower rate than our SA node. Sometimes we have problem with our SA node, and AV node can work as a pacemaker. But when the AV node become pacemaker, the number of heart rate decrease to 40 to 50 beats per each minute. It's fast enough to sustain life. But when the cardiac muscle heart rate become about 20 to 40 beats per each minute, it's fatal. And in this part, you need to answer these questions. But ionic events can create the depolarization phase of the SA nodal cells action potential, and what is the functional significance of the small size of AV nodal cells. Now, I want to talk about the cardiac muscle cells at rest. The cardiac muscle cells have a common membrane proteins. These muscles, we have sodium-potassium pump, we have calcium pump, and leakage channel for sodium and potassium. The cardiac muscle cell resting membrane potential is negative 90 millivolts. If you remember, in the nodal cell, the resting membrane potential is negative 60, and in our neurons, it's negative 70 millivolts. The cardiac muscle cell have a specific voltage-gated channels. Fast voltage-gated sodium channel, a slow voltage-gated calcium channel, and voltage-gated potassium channels. This is the cardiac muscle cell. In this cardiac muscle cell, you can see a striation, you can see branching pattern, and this is the cell membrane or sarcolemma, and you have different proteins. We have fast voltage-gated sodium channel, a slow voltage-gated calcium channel, and voltage-gated potassium channel here. I want to talk about the events which happen during action potential in our cardiac muscle cell. I want to talk about it on this slide. Look at this picture. The electrical events of cardiac muscle action potential are this. And you see that the graph is completely different from the graph of action potential which we have for nodal cell and for our neuron. The first step is called depolarization. When the impulse from the conduction system of the heart by the gap junctions reach the cell, the fast sodium-gated channels become open and sodium ion come to our cell very quickly, and it can make rapid depolarization of our cell due to sodium inflow when the voltage-gated sodium channels become open very fast. After that, we have a phase which we call it plateau phase. I forget to tell you that this depolarization continues until we reach positive 20 millivolts. 20 to 30 millivolts. It's different in different books. Then we have a plateau phase. The depolarization can open the calcium channels. The calcium channels are slow calcium channels. The calcium channels become open and calcium ion come to our cell. Look at here. This is the cell and the cell membrane. Calcium ion come to our cell, and at the same time, potassium ion leave our cell. So one positive ion inside, one positive ion leave the cell. So it means phase which is plateau. We don't have any change in the membrane potential. And after that, the calcium channels become closed and the potassium channels are still open. The potassium ion leave our cell and you can see repolarization of the cells. So in this graph, you can see the rapid depolarization due to sodium inflow when the voltage-gated sodium channels become open. Then we have a plateau phase when this calcium channels help the calcium ion come inside and potassium channels help the potassium ion go outside. And after that, we have repolarization. And during repolarization, you see that the potassium ion leave our cell and inside the cell become more and more negative, and again we reach the resting membrane potential which is negative 90 millivolt. And in this slide, you can see all electrical events which we have for our cardiac muscle cell. During depolarization, the fast voltage-gated sodium channels are open and sodium ion come to our cell until we reach positive 30 millivolts. Then the calcium channels are open, calcium ion come to our cell and potassium ion leave our cell, and this stage is called plateau stage. And then we have repolarization when calcium channels become closed and only potassium channels are open, and potassium ion go out of the cell and the membrane potential again reach negative 90 millivolts. Calcium in plasm from the interstitial fluid, and the sarcoplasmic reticulum start going to contraction, start the phase of contraction. Like the skeletal muscle, the calcium can attach to troponin and initiate the cross-bridge cycling. Do you remember the troponin has TNC subunit calcium attach on it, then the configuration of troponin change, it pull up tropomyosin, and then actin and myosin slide over each other. We have the same feature in our cardiac muscle, and the calcium level decrease help going to relaxation. Channels close and pumps move the calcium ion to sarcoplasmic reticulum and out of the cell, so we don't have enough calcium ion inside the sarcoplasm, and the actin and myosin go to relaxation phase. And cardiac muscle cells cannot exhibit tetany. In our cardiac muscle cell, unlike the skeletal muscle, we have a very long refractory period. I hope you remember the meaning of refractory period. Refractory period, which divides to absolute and relative. Refractory period are the period which we cannot have the other action potential. And the reason why do we have a long refractory period in cardiac muscle is this. The cell cannot fire a new impulse during refractory period. Cardiac muscle cell plateau phase helped a long refractory period, about 250 milliseconds. The heart cell contract and then become completely relaxed before the next stimulation start. And it can help our cardiac muscle not to go to the sinus phase. I complete this part for you, and we only have one other video and one other PowerPoint on heart physiology. I want to talk about cardiac cycle next time for you.